In this presentation, I will be talking about how genes are sequenced, and the syllabus points are: in 3.1, the entire base sequence of human genes was sequenced in the Human Genome Project, and in 7.1, the use of nucleotides containing dideoxyribonucleic acid to stop DNA replication in the preparations of samples for base sequencing. Now, the reason why these two syllabus points I'm covering now is because I feel that you need to have an understanding of the PCR method before you can truly understand this method of gene sequencing. So this was how genes were originally sequenced during the Human Genome Project using what's known as the Sanger sequencing method. Okay, so the key thing here is the use of these particular nucleotides. So this is dideoxyribonucleotide, and this is adenine. And what you can see here is normally you would have a hydroxyl group here. You would have a little O there, but it's not here, which means that when this is added to the growing strand of DNA, another one can't be added on here because there isn't an OH group for a covalent bond to form. So what happens is the DNA polymerase just falls off because it can't add any more nucleotides to that growing strand. So this is our dideoxyribonucleotide, or in this case, it's a nucleotide triphosphate, because they're the building blocks that are used to make DNA in DNA replication. Okay, so you've seen the little animation, I hope now, which I think is absolutely brilliant. In the animation, it does actually mention these, but it calls them terminator nucleotides, but we know they're dideoxyribonucleotides. Okay, so where are we? Okay, so how are genes sequenced? I'm going to give some summary notes here and just go through it. So we add standard deoxyribonucleotide triphosphates, are normal nucleotides with that OH group, and Taq polymerase are added to the DNA sample. And also you saw from the animation that you would add a primer. You just add the primer for one strand because you don't need to sequence both strands at once, because obviously one will be the complement. So you only need to really sequence one strand to know then the other strand. Small quantities, very small quantities of the dideoxyribonucleoside triphosphates are also added to the sample with different coloured fluorescent tags for each base. And then the process, very similar to PCR, is carried out. You have the heating and cooling cycles so that lots of different fragments can be formed. And because dideoxyribonucleosides have just a hydrogen instead of the OH on carbon 3, this means that during replication, mostly the standard nucleotides are added, but occasionally and at random, these dideoxyribonucleotides are added. And this means that replication is halted when the dideoxyribonucleotide is added to the strand. So what we get is fragments of lots of different lengths. So whenever that dideoxyribonucleotide is added, it actually stops replication and you get a fragment formed with a little fluorescent tag at the end of a different colour depending on whether it's A, C, T or G. And then the fragments can then be analysed. They're sent through a special sort of column form of gel electrophoresis, but it works in exactly the same way as normal gel electrophoresis. So these smaller fragments will carry first, the larger fragments will be, slowed down, and you'll get this sequence of bands, each ending in a different coloured tag depending on which base it finished with. And from that, you can analyse the final sequence of your DNA. Okay, so I won't do that. That's the notes that I want you to fill in. We just got to get through that. Okay, so this diagram shows what exactly is happening here. So this one was added fine, and then this one was added fine, but because it only has an H here, no more can be added. So this would create a fragment ending with our cytosine. So we know that at this point there's a cytosine. So that's how it works in terms of adding the dideoxyribonucleotides. And what you end up with is a little sequence like this, and you can see all the fragments. So the gel electrophoresis was carried out in this direction, so we know the smallest fragment at the beginning is A, then T, then G, then C, and so on. And you actually end up with a sequence that can be read fed by an automatic analyzer which will read those fluorescent sequences. And that is how genes are sequenced using the Sanger sequencing